A very big hello to all the CSIR NET Life Sciences aspirants. How are you all doing? In today's video, I'm going to discuss about Unit 3 important previous year questions. Unit 3, as we know, is the molecular biology part, and it is like in the syllabus, it is named as fundamental processes. Okay, so the topic that we are going to cover in today's video will be related to translation. So definitely we cannot discuss all the previous year questions related to translation in one video. So what I have done is I have taken the most important or the type of questions which has been asked repeatedly in the previous year question papers. Okay, and I have taken them and we will see how to solve them. And I expect that whatever I tell you today in today's video and whatever tips I will give you, that will help you all in solving the previous year questions similar to that. So starting with the very first question. Now this question is about amino acyl tRNA synthesis. Okay. Now, if you have gone through the previous year question paper, you will see like last 5-year question paper. If you see every session, almost every session, at least one question has come related to amino acyl tRNA synthesis. Okay, so this topic from this translation has become a very important topic. Okay, so what is amino acylation of tRNA? How the amino acylation of tRNA is done? How the tRNA is recognized by amino acyl tRNA synthetase? If there is wrong amino acylation, how it is corrected? So you have to study each and everything about amino acyl tRNA synthetase. Okay, so you can see it is a statement based question. It's a fact based statement based question which you will be able to answer very easily if you know these things about amino acyl tRNA synthetase. Okay, so you have to read everything that is available in a standard book about this particular enzyme, because it is one of the very important topic for CSIR NET Life Science exam. So let's see this question. Fidelity of protein synthesis depends to a large extent on the accuracy of amino acylation of tRNAs with correct amino acid. However, given that the side chains of many amino acids are not sufficiently different, amino acyl tRNA synthetases are often prone to misacylate the tRNA. Misacylating tRNA means attaching a tRNA with a different amino acid from the one that it is supposed to attach. So one such example of misacylation is by tRNA threonine. Okay, so threonine, which the tRNA that has the anticodon for threonine, by threonyl tRNA synthetase. Now, in this context, following statements are being made about E. coli threonyl tRNA synthetase. Okay. See, so what background information you have to know? That first of all, yes, it is true that the fidelity of protein synthesis depends on amino acyl tRNA synthetase, because amino acyl tRNA synthetase is the one which is going to attach the amino acid to its respective tRNA. Okay, so this process is catalyzed by amino acyl tRNA synthetase. Okay, and then after that it becomes the amino acyl tRNA. Okay, now in the cell there are specific amino acyl tRNA synthetases, each for a different amino acid. They are specific towards their amino acid. Okay, now we know that there are certain amino acids where the side chains are not very different from each other, like leucine, isoleucine, or serine and threonine. Okay, so like this, there are certain pairs or certain groups of amino acids which has similar side chains. Now, when they have similar side chain, then the chances are high that the amino acyl tRNA synthetase does a misacylation. That is, it does a mistake. Okay, so suppose tRNA was supposed to be attached with amino acid 1, but the amino acyl tRNA synthetase has done a mistake and misacylated the tRNA with amino acid 2. Okay, so here we are talking about the threonyl amino acyl tRNA. So statement 1 says it misacylates tRNA threonine equally with serine and cysteine. Okay, so this statement is wrong. Till serine it is correct. So serine and threonine, they have similar side chains. So yes, threonyl tRNA synthetase, it misacylates the tRNA for threonine with serine. Okay, but it does not do it with cysteine. Okay, so that's why this statement becomes incorrect. Second, it possesses a distinct editing site that preferentially deacylates the misacylated tRNA threonine. Okay, now that is actually true. When we see the structure of amino acid tRNA synthetase, so just roughly if I draw, so it will have two sites. Okay, if this is the active site, so active site is the site that will be used for amino acylation. Then there is the editing site. Okay, so if any mistake is done, or if any misacylation by that particular tRNA synthetase is done, then this editing site, okay, which is present in the same amino acid tRNA synthetase, will correct the misacylation. Okay, so it possesses a distinct editing site that preferentially deacylates the misacylated tRNA. This is true. C statement, editing of misacylated tRNA threonine occurs frequently in cis before the release of misacylated tRNA threonine. Okay, yes, that is again true. So here only, so where if this amino acid tRNA for threonine, okay, got attached with serine, okay, then this editing site will immediately remove the threonine. Okay, so it will release the serine, okay, and it will make sure now that threonine is added. Okay, so editing of the misacylated tRNA threonine occurs at the cis before the release of misacylated tRNA threonine. Okay, so misacylated tRNA will not be released. Okay, so as soon as the misacylation done, it will be detected by the editing site. It will correct the misacylation, and then only the tRNA will be, the amino acid tRNA will be released. Okay, so this statement again is true. D, it possesses a distinct editing site that does not discriminate between misacylated tRNA threonine and threonyl tRNA. So this is wrong. It does discriminate. If it discriminates, then only it will be able to edit, right? So this statement is wrong. And statement E, the amino acylation of the editing site of amino acylation and the editing site of the threonyl tRNA synthetase are same. Now this is wrong because it is not same, it is different. There will be two sites present in amino acid tRNA synthetase. Okay. Now you have to remember one more thing, that this editing site is not present in all the amino acid tRNA synthetases. It is present in only those amino acid tRNA synthetases where there is chance of doing a misacylation. Okay, for the amino acids with similar side chains. Okay, those amino acid tRNA synthetases only will have this editing site. So choose the option that represent all correct statements. So the correct statements are B and C. So we have our option as option 2. Next type of question which is asked from translation is about the translation inhibitors. Okay, so this question is asking about chloramphenicol. So whatever translation inhibitors are there, so whether it is chloramphenicol, whether it is streptomycin, whether it is tetracycline, whether it is diphtheria toxin, so all these translation inhibitors and their mechanism of action, you have to know. Okay, so what you can do when you are studying is you can make a table of all the antibiotics and their mode of action. Okay, not only antibiotics, the translation inhibitors. Okay, because part B as well as part C type of question has been asked. Chloramphenicol is a broad spectrum antibiotic which inhibits protein synthesis in prokaryotes. Given below are a few statements regarding the mode of action of chloramphenicol. So chloramphenicol inhibits the peptidyl transferase activity of ribosome. Chloramphenicol can be used to treat moderate to severe infections because mitochondrial ribosomes are not sensitive to chloramphenicol. Chloramphenicol binds to one of the domain of 23 srRNA. Chloramphenicol competes with binding of E-site tRNA. Which of the following options describe correctly the mechanism of action of chloramphenicol? So what does chloramphenicol do? So basically when chloramphenicol is there, okay, so this is the mode of action. So when chloramphenicol is there, it stops the peptidyl transferase activity. Okay, so if it will stop the peptidyl transferase activity, because of which what will happen? Peptide bond will not form. Okay, So chloramphenicol inhibits the peptidyl transferase activity of ribosome. This statement A is correct. Now, how does chloramphenicol do it? Because chloramphenicol binds to one of the domains of 23S rRNA. Okay, so it is in the 23S rRNA of the larger ribosomal subunit which has the peptide bond, this peptidyl transferase activity. Okay, so chloramphenicol binds to one of the domain of 23S rRNA and it will stop the peptide bond formation. So this statement C is also correct. So answer is A and C only. All right. One last type of question that can be asked from translation is related to this kind of a question where the data is given, this kind of analytical type of question where the data is given to you either in the form of a table or in the form of graph. Okay, this is a typical analytical type of question which can be asked here where there is some experimental condition is given and you have to analyze, you have to understand the situation and correspond it with the graph that is given here to find the correct answer. Okay. Now let's see this question. So ribosomes prepared from a bacterium were fractionated by sucrose density gradient centrifugation to separate 30S, 50S and 70S population. Okay. So this is panel 1. So panel 1 is the one where we have isolated ribosome from bacterial cell and then separated the different components. Okay. So we see that, so this peak is for 30S, the smaller ribosomal subunit, this peak for largest ribosomal subunit, and this is for the holo ribosome, okay, which is a combination of 30S and 50S, okay. So these peaks here, what are they representing? They are representing the concentration. Now, when the ribosome preparation was incubated individually with either elongation factor G or a newly identified protein X or GTP, the profile remained unchanged. Okay. So what it is saying, I'm just going to make a table of what the information is given here. So it is saying that when the ribosome preparation plus elongation factor G, okay, or ribosome preparation plus a new protein that has been isolated, which is X, or the ribosome preparation with GTP. Okay. In all the three cases, there is no change in the profile. The same, this profile only it is given. Okay. Now, next it is saying that likewise, no changes were seen in the profile. So again, there is no changes in the profile when ribosome preparation was incubated with EFG plus GTP or protein expert plus GTP. Okay, so basically here in this first set, we have used all the components one by one. Now we are going to use them in combination. Okay, so what is the combination? Ribosome preparation plus EFG plus GTP. Okay, and also ribosome preparation plus protein X plus GTP. And in this situation also, there is no change. Okay, same graph one only we are getting. Okay. However, when the ribosomal preparation was incubated with X, EFG and GTP all together, it resulted in the change in the profile which showed a decrease in 70S peak area and increase in the peak area of 30 and 50. So you can see here the second panel. So when are we getting the second panel? So I'm writing it here only right away. So the second panel we are getting when there is ribosome preparation is incubated with EFG plus protein X plus GTP. Okay, then we are getting this particular profile. All right, and in this particular profile, what is the situation? That the peak of 70S has gone down, whereas the peak of 50S and 30S has increased. Okay, now if this is just the graph you have, okay, so just looking at the graph, what you can conclude that 70S concentration is decreasing compared to the first graph and 50s and 30s concentration is increasing, right? So when this can be possible, it is possible when the 70s is breaking down into 30 and 50. So 70s, when it is breaking down into 30s and 50s, then only this situation is possible where the concentration of 70s is decreasing and the concentration of 50s and 30s are increasing. Okay, right? Now, how can a 70s concentration decrease? If there is any dissociation factor, means there is something which will dissociate both the subunits. Okay. Now, what is the question? Choose the option that defines the correct conclusion from the observation. Protein X is an anti-association factor which functions in the presence of EFG and GTP. Now, this is sure that protein X only is causing something, and protein X can work only in presence of EFG and GTP both. Okay, because protein X, when it was used alone or it was used just with GTP, there is no change. Okay, so for proteins X function, it requires EFG plus GTP. Okay, now the question is whether it is an anti-association factor or it is a dissociation factor. See, if this would have been an anti-association factor, then protein X will stop the formation of any new 70s. Okay, but here it did not happen like that. Instead, what was happening? Whatever was already there, whatever 70s component was already there in the first profile, that was broken down into its subunit, which is possible when protein X is a dissociation factor. Okay. So, if this was an anti-association factor, then in that case, the profile again will remain same because it will not allow 30 and 50s to combine and form 70s. Okay, so 70s concentration will not let it increase. But here it is not the situation. The situation is 70s concentration is decreasing. Okay, that means there is some component which is breaking down 70s into its subunits, and that has to be protein X. And we saw in the experiment that protein X alone cannot work. It cannot even work just with GTP. For its work, it requires EFG plus GTP. Okay. So option C says protein X binds to GTP. That there is no conclusion of that. And D says that EFG is known to bind GTP. Hence, it can be concluded that the effect of GTP is through EFG and protein X does not bind GTP. Okay. Now, there is no conclusion of this as well. Okay. So, the most probable conclusion, most correct conclusion from the observation will be that protein X is a dissociation factor and it works in presence of EFG and GTP. Okay. So with this, we come to the end of this video. I hope you found it informative, you found it helpful. Please use the same concepts and try and solve more questions from translation. Thank you everyone. See you again in another.